I collected my data through three independent viability assays, first of which is called an MTT assay. Basically, MTT is a tetrazolium dye, which is a yellowish color, and when you put it in a solution full of cells, it will be reduced by the cell's reductase enzymes to a formazine, which gives a purple color. And so you measure the amount of purple using a plate reader, and then you quantify that, and it's directly proportional to the number of living cells. The second assay I used also involved a plate reader, but this time it was reading for luminescence. And this luminescence assay relies on the leucophase enzyme, which is found in fireflies, and it takes the ATP of the living cells and it generates light energy, which can be measured and is proportional to the number of living cells. And the primary difference between the MTT and ATP assay I'll go over later, but the ATP assay gave me a wider range and was more precise in that light. And so finally, after my third assay involved, after I put the soap on the E. coli, I pipetted the remaining solution onto an agar plate and left them to grow overnight in an incubated chamber. And so I measured the density of the colonies the next day, which sort of served as a confirmation to my other quantifiable data. So I'll be discussing a bit of each assay in detail. So first of all, MTT. MTT is a sort of the standard viability assay. It's widely used to measure in vitro effects of drugs on various cell lines and in primary patient cells. However, according to studies done, it can also be effective in measuring bacteria viability. Basically, this goes over how it works again, and the metabolic activity which we measure is proportional to the number of viable cells. And so this should offer a very good comparison point between the three disinfectants that I will test. For the ATP-based luminescence, I used a CellTiter Glow Cell Viability Assay Kit, which is also a homogeneous method to determine the number of viable cells. And I selected this assay because it's stable even in the presence of abnormal pH and detergents, which makes it an ideal assay for this experiment. In addition, data can be collected in just over 10 minutes, and the half-life of the signal lasts 5 hours. So it's very simple in its implementation, and is very effective and efficient. Moreover, this assay provided me with a wider range since we were measuring luminescence, and so the distinction between the three disinfectants I tested was clearer. So finally, my third assay was not quantifiable, but it was sort of a backup assay in that there is very low chance of error, and it also served as a confirmation to my results. So the process I took was, after I applied the soap to the bacteria, I would mix the solution and then pipette a portion of that, 100 milliliters, onto a petri dish containing LB agar. And then this contains all the nutrients that bacteria need for growth. So overnight in an incubated chamber, if there was any bacteria left alive after being in contact with cleansing reagents, they would grow, and I would be able to differentiate the disinfectant capabilities of each disinfectant by looking at the densities of the bacteria growing on the plate. And so I did this on a scale plus, minus, and 0, which in words, that's plus relates to the colonies being too dense to differentiate. Plus is it's very heavy growth, but you can differentiate colonies. Minus would mean sparse growth, and 0 would mean no growth, which indicates that all the bacteria were killed. And so that looks like this. So um, the pictures are labeled, but from right to top left would be plus plus. You can see that the whole petri dish is covered in bacteria, and it's very hard to differentiate between the colonies. Plus would be on the top right, and they look very similar. However, you can clearly differentiate between the glinting colonies in the top right. And minus was also very clear in that there's very little colony growth, but there are bacteria growing. And 0 was very easy to differentiate from the others because the plate remained clear. So transitioning from design into results, I'll first start with my raw data, which will go through each of the cleansing reagents separately. So first of all, you can see that bleach was extremely effective, and if you remember, I tested from 3.23% to 25% in my design. I designed my experiment to do that. However, bleach was so effective that to generate a curve, I had to go lower in concentration. So that's what you see in the first 3 data points. 0.41% on average killed 32% of the bacteria. And you can see that bleach is extremely effective, even at 1.64%, killing 90% of the bacteria. And so, moving on to hand sanitizer, we don't see the same trend. In fact, I think the best way to look at this is to look at the colony growth on the agar plates and compare those. So in bleach, they stopped growing on agar plates, meaning that virtually everything was eliminated at 3.23%. However, comparing that to hand sanitizer, we can see that it's not nearly as effective. And however, we also do see that as concentration goes up, hand sanitizer becomes more and more effective. And finally, we have the antibacterial hand soap, which in all trials had very dense growth, led to dense growth on the agar plates when grown overnight, and according to the readings, killed no more than 60% in its highest concentration. So what that looks like visually in a graph, as you can see, bleach was consistently the best disinfectant, killing over 99% in every trial, starting from comparing to the other 2 reagents. Hand sanitizer became more and more effective as the concentration increased, almost to the point of bleach. So at 25%, it was nearing the level of bleach, killing about 90%. And hand soap, although it gradually increased, was never able to kill more than 65% of the bacteria. And so as a result, all the plates with hand soap applied showed dense colony growth when seen the next day. And so my results led me to 3 conclusions. And as I've said, hand soap is very limited as a disinfectant. And on the other hand, hand sanitizer became more and more effective as concentration increased, and bleach was an extremely effective sterilizer. So the method I used in this investigation I viewed as successful, and the results generated corresponded well to both my hypothesis and similar experiments done. However, there are several improvements that could be made. The first of which is another parameter, which would be time. In testing the effectiveness of a cleaning agent, it's also important to consider efficiency. And so, if I were to do this experiment again, I would add a parameter of contact time for example, ranging from 5 minutes to 25 minutes in increments of 5 minutes. This would expand the scope of my investigation, but also add depth as well. And another reason why time is very important is because research has found that the antibiotic in hand soap actually increases in its effectiveness over time. So it'd be very interesting to investigate this. The second improvement that could be made is to use mixed culture instead of just E. coli. This experiment centered around one strain of bacteria, and while the results were clear, it would add thoroughness to my investigation if many different strains were tested in a mixed culture. And so this could be done by experimenting with bacteria collected from an aerobic sludge sequencing batch reactor, which is found in many labs, and this contains many strains of bacteria found in wastewater. Finally, there was one limitation which I noticed after collecting data in the MTT assay. In contrast to eukaryotic cells, for which MTT is widely used as a viability assay and consistently provides very thorough and stable results, E. coli and bacteria in general lack the mitochondria and instead generate ATP through cytosolic processes. And since the data generated from the MTT assay relies on the presence of reductase, its sensitivity to measuring the viability of bacteria and E. coli may be severely impacted due to the absence of large quantities of mitochondria. And this was noted in my experiment in that there was a clear line of background, and it was very hard to differentiate the MTT readings from each other. So, in that sense, the luminescence assay which I used provided clearer distinctions and better data in general. This concludes my presentation. Thank you for listening, and I hope you enjoyed and learned as much as I did.